So yeah, good morning, everyone. Let's go ahead and get started. Are there any questions before we begin today?
I sent you an email. Do you prefer Canvas or should I send it a different way for Vanessa?
I can respond to your email later.
Okay.
Was it urgent?
It's I didn't know how to get to my what bio class for the lab I was supposed to do, so I didn't do any last week. I didn't
Okay.
know I had to go in person because Canvas and my schedule say two different things.
Okay, yeah, I'll get back to you through email.
Thank you.
Is there any other questions? Okay, so today we're going to talk about exploring life and science, the characteristics of life. And so the learning outcomes for today is to explain the basic characteristics that are common to all living organisms, describe the levels of organization of life, summarize how the terms homeostasis, metabolism, development, and adaptation all relate to living organisms, distinguish between science and technology, and then summarize some of the major challenges facing science today. Okay, so first off, life is organized. So we have different levels of biological organization. So we start with an atom, and with atoms, we can build larger molecules, larger macromolecules, and then these large macromolecules make up cells. And cells are the smallest structural unit and functional unit of an organism. Okay, and so some organisms are single-celled, like bacteria. However, most animals are multicellular, like humans, which are composed of many cells. And then once you have cells can start to build up tissues, which are groups of similar cells that perform a particular function. And then from tissues, you can build organs. So organs are composed of several tissue types. And then an organ system composed of different organs work together for a common purpose. And then from an organ system, you get an organism. Okay, and that's the collection of all the systems inside the body. And from organisms, we get groups of species that interbreed. And then different species interact with other species. And you get a community, and you have populations as well. Okay. If you zoom out a little bit farther, we get an ecosystem, which is the community of populations interacting with the physical environment. And then lastly, you get the biosphere, which is all of the Earth's ecosystems. Okay, so life requires materials and energy. So energy is the capacity to do work. And humans acquire materials and energy by eating. Okay, so food provides the nutrients, which are used for building blocks and energy. And that energy is usually made through metabolism. So metabolism is all of the chemical reactions that occur within a cell. And the ultimate source of energy for life on Earth is the sun. So all the energy that you get essentially came from photosynthesis, which is used by plants, algae, and some bacteria to harvest energy from the sun and convert it to chemical energy. And so what photosynthesis does is it produces sugars, which serve as the basis for the food chain of other organisms. Okay, another vocabulary word that's important here is homeostasis. So living organisms maintain an internal environment that's different than the external environment. Okay, so most organ systems strive to maintain homeostasis. For example, body temperature is maintained within a narrow range of values. And if you're out of homeostasis, you might have a temperature or a fever. Okay, so living organisms also respond to external stimuli. So homeostasis would be impossible to maintain without the ability to respond to stimuli. For example, removing your hand from a hot stove or an internal stimuli, something like adjusting your blood pressure in response to values outside of normal. So another thing that organisms do is they reproduce and develop. So when organisms reproduce, they pass on their genetic information to the next generation. So first off, they need to grow or go through growth, which is the increase in size and the number of cells. They also need to go through development. And that's basically all the changes that occur from fertilization until death. So these include changes during childhood, adolescence, adulthood, and also repair after injury. Okay, and if you don't remember, the basic genetic information of all life is DNA, or deoxyribonucleic acid. And this molecule contains the hereditary information that directs the structure and functions of all cells. Okay, and inside DNA, there's genes, these short segments that specify traits. And you can have mutations in these genes, so there's variation. So an example is like hair color, right? So there's genes that can control hair color and height, things like that. So these can be beneficial and make an organism better suited for its environment. And this is the basis for evolution, that there's mutations in genes and those genes get selected for. Okay, so that's the term evolution. So evolution is how a population changes over time. And this occurs through a process called natural selection. So this is what we say, the process by which evolution occurs. When a new variation occurs that allows organisms to capture more resources, those individuals have more offspring. And we call this an adaptation. So over time, population has more individuals with this advantageous variation. So just a little check-in for the first part. You want to be able to list the basic characteristics of life. You want to be able to summarize the levels of biological organization. And you also want to be able to explain the relationship between adaptations and evolutionary change. Is there any questions so far? Okay, so another thing that you need to know is that humans are related to other animals. So the learning outcomes here are to summarize the place of humans in the overall classification of living organisms, understand that humans have a cultural heritage, and describe the relationship between humans and the biosphere. Okay, so first off, all life is classified into one of three domains. So we have bacteria, archaea, and eukarya. So bacteria and archaea contain prokaryotes, and prokaryotes are single-celled organisms that lack a nucleus, where eukaryotic cells contain a nucleus, where some eukaryotic cells can be single-celled as well, but most are multicellular like humans. Okay, and what's important here is that evolution requires time. So if you look down here at the bottom, this is showing you the relationship of the three domains of life. And you can see that this scale here is billions of years. Okay, so it takes billions of years for new species to evolve. And what it's showing you here is that the common ancestor was some first cells that we don't really know how life evolved on Earth or if life did evolve on Earth, but we know that there were some common ancestors, some first cells that ended up on Earth. And from there was a branching point into bacteria and archaea. And from archaea, there was another branching point for eukarya. Okay. And in eukarya, there's four kingdoms. So we got the protists, plants, fungi, and animals. And kind of what this is showing you is that animals are more related to fungi than animals are to protists, archaea, and bacteria. Okay. And bacteria and archaea are thought to be really old species. And in fact, there's about like 1 trillion different bacteria species, many more that need to be found. So if you're interested in microbiology, it's definitely something that's interesting there. Okay, and then like I said, the domain Eukarya is divided into 4 kingdoms. So we have plants, fungi, animals, and protists. Okay. So for archaea, these are prokaryotic cells of various shapes. And most of these are extremophiles, so they can survive in extreme temperatures. I think they can survive up to 110 degrees Celsius, which is boiling water. So you find these in hot springs and things like that in Yellowstone. Okay. They absorb or chemosynthesize food, and they have unique chemical characteristics. And something to note here is the magnification here. This is at 33,000x. So these things are very small. You need microscopes to see them. Okay. Domain bacteria, these are also prokaryotic cells of various shapes. They're very adapted to their environment. They've been around for a while. And they can absorb, photosynthesize, or chemosynthesize food. And they have unique chemical characteristics. And then again, look at the magnification here. This is at 6,000x. So again, these things are very small. This is an example of E. coli. Okay, and then moving on to kingdom Protista. So these are your algae, your protozoans, your slime molds, and your water molds. So these are complex single cells. Sometimes they exist in filaments or colonies, or even multicellular. And these also can absorb, photosynthesize, or ingest food. So this is an example of a paramecium here. It's a single-celled protozoan. Okay, kingdom Plantae, so we're all familiar with plants. This is an orchid here. So we got our algae, our mosses, our ferns, conifers, and flowering plants. Most are multicellular, usually with specialized tissues containing complex cells. And then if you forgot, plants also photosynthesize food. Okay, kingdom fungi. So these are your molds, your mushroom, your yeast, and ringworms. These are mostly multicellular filaments with specialized complex cells, and they also absorb food. Okay, and then the last one here is kingdom Animalia. So there's tons of animals, there's millions of different species. So we have sponges, worms, insects, fishes, frogs, turtles, birds, and mammals. Again, these are multicellular with specialized tissue containing complex cells. And these animals ingest food. They're not able to create their own food. Okay, and then in kingdom Animalia, there's 2 separations. Most organisms in kingdom Animalia are invertebrates. So these are your earthworms or insects or mollusks. And then the rest of them are vertebrates, which have a nerve cord protected by a vertebral column. So these are your fish, your reptiles, amphibians, and birds. And vertebrates with hair or fur and mammary glands are called mammals. So humans, raccoons, and seals, things like that. Okay, for humans, humans are most closely related to apes, but are distinguished from apes by a couple different things. So 1, they have a highly developed brain. Number 2, completely upright stance. Number 3, creative language skills. And number 4, ability to use a wide variety of tools. Okay, so what's important here again is that humans and apes have a common ancestor. Something else that's important here is that humans have a cultural heritage. We're the only species that does. So culture are activities and items passed down from one generation to the next. So these are beliefs, values, and skills, and arts and sciences. Okay, so any questions about species? Okay, so moving on to science as a process. So the learning outcomes here is for you to describe the general process of the scientific method, to distinguish between a control group and an experimental group in a scientific test, recognize the importance of scientific journals and the reporting of scientific information, interpret information that is presented in a scientific graph, recognize the importance of statistical analysis to the study of science. Okay, so science is a way of knowing about the natural world. And as scientists, we try our best to be objective or factual and not subjective. So subjective information usually involves some sort of judgment. Okay, so we collect objective or facts through the scientific process, which is also called the scientific method. Okay, and so there are specific steps for the scientific method. Step one is we start with an observation. Number two, we develop a hypothesis about that observation. And we use inductive reasoning. So inductive reasoning is defined as someone using creative thinking to combine facts into a cohesive whole. Okay, and the scientific method isn't pretty, it's a messy process. So you'll start with a hypothesis, you'll do an experiment, and then from that experiment, you'll have more hypotheses, and you'll have to run more experiments. So you can see in this example, there's 6 different experiments that have been run to finally get a conclusion. Okay, so there's a lot of back and forth between the drawing board and trying to figure out what's going on, and then designing your experiment to test your hypothesis. Okay, so steps of the scientific method: you make a prediction and perform experiments. And like I said, you're probably going to perform lots of experiments. Scientists use deductive reasoning to test the hypothesis, if-then logic. Good experimental design includes an experimental variable and responding dependent variables. Also need to include test groups and a control group. Test groups are exposed to the experimental variable. The control group is not. And in science, we often use models, like model organisms like mice or C. elegans or Drosophila, to control for variables. Okay, after we've come up with our experimental variables and our control group, we collect and analyze data, and we report that as results. So statistical data includes a standard error or a standard deviation to show how uncertain the value is. So a lot of times in science, we're using a machine to collect the data for us, and those machines have some sort of standard error, which they report in their graphs. So I'll show you that in a sec. If you see data published in a scientific journal, that means it's been peer reviewed. Okay, and then lastly, we develop a conclusion. So we analyze the data in order to reach a conclusion about whether a hypothesis is supported or not. And like I said before, the conclusion of one experiment can lead to the hypothesis for another experiment and another experiment, and so on and so forth. Where at the end of the day, you're doing dozens of experiments to test your whole hypothesis. Okay, so the presentation of scientific data. So we usually report in graphs and tables and things like that. So here is showing you the y-axis is showing blood cholesterol levels, and the x-axis is showing you over time. Okay, so time would be the independent variable here, and the cholesterol would be the dependent variable. And you can see how this patient has a variation in blood cholesterol levels as the week goes on. So from this data, you can kind of hypothesize new things, like maybe they ate McDonald's during week 2 or something. And then what I was saying earlier about the standard error is how the machine or whatever you use to collect this data is reporting this value within this range. That's what we call the standard error. Okay, and so after the scientific method, you develop your hypotheses and your conclusions, you eventually get to a scientific theory, which is an accepted explanation for how the world works. So we have cell theory, for example, which is how all organisms are made of cells. Okay, so other than scientific theories, we have laws or principles, which are accepted by an overwhelming majority of scientists. So for example, like evolution. Okay, so just to give you an example of a controlled study, say for example, you're testing different antibiotics and you want to know which antibiotic is better at killing off bacteria. So you come up with a hypothesis, which is that antibiotic B is better than the currently used antibiotic A. Okay, so first you need to establish a control group. So a control group will receive no antibiotics at all. So subjects with ulcers, so this is checking for ulcers. So subjects with ulcers receive a placebo. So that's a pill that contains no medication. And then you come up with 2 test groups and each receive one of the antibiotics. And what's important here to do is a double-blind study where neither the doctors nor the patients know which group they are in. Okay. And from conclusion, we see that antibiotic B had better results. And I'll show you that here. Okay, so again, we state a hypothesis. Antibiotic B is a better treatment for ulcers than antibiotic A. We have our individuals that we're going to test. We split them up into 3 groups. We have our control group that received placebo. We have group 1 that receives antibiotic A. And then we have test group 2, which receives antibiotic B. And then you collect the data. So here's a doctor here, actually collecting the data, looking for ulcers. And then you report that as a graph. So as data. So effectiveness of treatment. So again, on the y-axis, we have % treated. And on the x-axis, we have % of ulcers cured. And you can see in the control group, there was very little that were cured. Test group 1B had some effectiveness, but group 2, you can see that 80% of the treated were cured. So from this data, we can conclude that test group 2 or the antibiotic B is a better antibiotic. Okay, any questions about that? Okay, so the last topic here is challenges facing science. So one of them is technology. So technology is always going to grow. And as scientists, we always want to use the highest technology in the lab and in the doctor's office, things like that. So we're always struggling to keep up with that. So the application of scientific knowledge to human interests, right? So we always want to keep human interests at the top. Another challenge is biodiversity. So the total number and relative abundance of species, the variability of their genes, and the different ecosystems in which they live. So we're very aware of extinction, so the death of a species. And we're very alarmed at the current high rate of extinction. Okay, so that's something to always be aware of. Another frightening thing is the amount of emerging diseases, especially right now with the COVID going on. So some come from new or increased exposure to certain animals. Some come from globalization, so they spread more easily. And some diseases mutate and change hosts. Another challenge is climate change. So changes in climate due to human activity. So we're all well aware of global warming, an increase in temperature due to increase in carbon dioxide levels in the atmosphere. Okay, and just to check your progress for that section, explain how a new technology differs from a scientific discovery, explain why the conservation of biodiversity is important to human society. And lastly, summarize how emerging diseases and climate change have the potential to influence the entire human population. Okay, and that's it for today. Was there any questions about the material? Do you guys have any questions?
I think we're good. Thank you.
Okay. Sounds good.
Mr. Hagee,
Yeah.
I had a question. About the McGraw Hill Connect. I was gonna ask if we had if you knew the access code or if we had to buy it.
Yeah, you're gonna have to buy that. So make sure you guys, when you're doing the Connect, that you go through Canvas. So when you're on Canvas, let's see if I can pull it up. So when you're on Canvas, you want to click this little McGraw Hill Connect link down here, and it should say sign up or go to Connect. And if you just click on that, it should take you right to Connect. And for this week, you guys do have chapter 1 and chapter 2 assigned for the IRAT and the Smart Book. Okay. Is there any other questions? All right, we'll wrap it up there and I'll see you guys next time, okay?
Thank you. Have a good day.
You as well.
Thank you.
Thank you.
Bye.